welcome to another Explainer. Today, we're going to unpack the absolutely incredible, complex biological journey of light turning into the images we consciously experience. Seriously, if you've ever stopped to think about how you're actually seeing the screen right in front of you right now, well, you're engaging with one of nature's most fascinating creations. Let's get right into it. So, did you know your eyes literally project the world completely upside down onto the back of your head? I know, it sounds crazy, but let's dive right into this mind-bending fact by tracking a single bounce of light from an object, say a dog, all the way into your brain. Because vision doesn't actually start in the brain, right? It starts out in the world, the very second light reflects off a surface. And we're going to follow that exact path today. So here's the roadmap. Step 1, the cornea. Step 2, the pupil. Step 3, the lens. Step 4, the retina. And finally, step 5, the brain. We're looking at a continuous path of light moving from the transparent front of your eye straight back to your visual cortex. It's this wild journey of refraction, chemical cascades, and electrical impulses. And get this, it all happens in a fraction of a millisecond. Let's break down exactly how each of these steps functions. Section 1. Light enters the eye. Our story starts the exact millisecond that light makes contact with the outer surface of your eye. Now, the eyeball itself is kind of soft and vulnerable, but it's protected by your hard skull, orbital fat, and that tough outer white layer known as the sclera. But to actually let light in, the very front needs to be perfectly clear. Enter the cornea. You can really think of the cornea as a protective clear window that does the heavy lifting of bending the light inward. Because it's curved and completely transparent, the second light hits it, its direction changes just a little bit. This right here is the very first stage of focus, grabbing all that scattered light from our environment and aiming it directly into the eye. Now, just past the cornea is the colorful iris. And the iris acts exactly like an automatic camera shutter, shrinking or expanding the pupil depending on your environment. Think about it. If you step into a dimly lit room, muscles in the outer area of the iris contract to dilate the pupil, letting more light in, right? It smoothly floods your eye with whatever light is available. But step out into the bright sun, and those inner muscles instantly pull the pupil tight to limit that light intake. It's a rapid adjustment, and it's absolutely crucial for protecting the sensitive hardware deeper inside. Moving on to Section 2, the lens in action. So after passing through the pupil, the light hits this incredibly dynamic, adaptable piece of biological hardware, your lens. While the cornea does the initial bending, the lens is responsible for the fine-tuning. The really crucial point here is that your lens physically deforms. It widens for distant objects and it bulges out for near objects right in front of you. This process is actually called accommodation. See, the lens is attached to these tiny ciliary muscles by microscopic threads called zonules. When you look at something far away, like a distant mountain, those muscles relax, pulling the lens nice and thin. But when you look at something up close, like your phone, the muscles contract. This allows the lens to shrink and bulge, perfectly focusing those scattered light rays onto the back wall of your eye. But what happens if things aren't perfect? Well, if your eyeball is shaped slightly too long or too short, the lens just can't perfectly place that focal point on the back of the eye. And that is the physical reality of being nearsighted or farsighted. For instance, if your eyeball is too long, which causes nearsightedness, the light converges into a focal point just in front of the retina, resulting in a blurry image. If it's too short, the focal point falls behind it. Things like glasses or contacts, they simply add an extra layer of refraction to correct that specific physical misalignment. Pretty neat, right? All right, Section 3, the retina and photoreceptors. Let's move to the back of the eye and see how this focused light finally becomes a biological signal. The back wall of the eye is lined with a thin, incredibly complex layer called the retina. And guys, this right here is where physics literally turns into biology. What's truly staggering about the retina is the sheer scale. We are talking about 130 million microscopic, light-sensitive cells lining your retina, just waiting to catch that incoming light. 130 million. And to even reach these cells, the light actually has to pass through several transparent layers of nerve fibers and specialized biological tissue first. So what are these cells? Well, we've got rods and cones. We rely on the extremely numerous rods to see in dark, shadowy conditions. The cones, on the other hand, detect bright light and colors. There are actually 3 types of cones tailored specifically for red, blue, and green light. Now, these cones are densely packed into a central area of the retina, highly concentrated in a tiny pit called the fovea centralis. This is the exact spot responsible for your absolute sharpest, highest definition vision. Rods are spread out more towards the edges, which is exactly why your peripheral vision is great for detecting motion in the dark, but, as you've probably noticed, totally terrible for reading text. Okay, so translating this light to electricity. When photons hit these photoreceptors, it actually alters their resting electrical potential of -70 millivolts. This kicks off a super rapid chemical cascade. First, horizontal cells coordinate the signals for high contrast. Then, bipolar cells step in to amplify or inhibit those impulses. Next up, amacrine cells analyze the incoming data before finally passing the baton to the ganglion cells, which transmit the final signal to the brain. It's basically a biological supercomputer processing the image before it even leaves your eye. But here's a crazy little flaw in the system. Because the optic nerve needs a physical place to exit the eye and travel to the brain, every single human has a literal blind spot. I mean, an exact area containing absolutely zero rods or cones where no light can be detected at all. This area is called the optic disc. You are entirely blind in this tiny patch of your vision, but you never notice it. Why? Because your amazing brain is constantly just filling in the missing data based on the surrounding environment. Which brings us to Section 4, the upside-down world. Guys, this is the biggest biological plot twist in our entire story of vision. Because of the way the lens refracts light rays, the image projected onto your retina is completely upside down. Due to the physical laws of refraction, by the time those light rays pass through the curved cornea and the lens, the image cast onto the back of your eye is completely inverted. It is absolutely wild to think about. The light bouncing off the top of a dog's head actually lands on the very bottom of your retina, and the light from its paws lands right at the top. No way, right? So why isn't the whole world upside down for us? To understand that, we have to look back at the 1930s. Scientists ran these famous Innsbruck goggle experiments to test how our brains handle inverted vision. Participants wore goggles that completely flipped their vision. Basically, the goggles forced the light to land on the retina right side up, which meant the brain was suddenly receiving upside-down data compared to what it was used to processing. You can only imagine the utter chaos that followed. Looking at the timeline for this experiment, the initial days were rough. Participants stumbled around, literally trying to step over ceiling lights because they looked like they were on the floor. But here's where it gets incredible. By day 5, their navigation and motor performance drastically improved. With continued exposure, their brains naturally corrected the input. The brain essentially reflipped the image so they could function normally again. Now, vision scientists note that the brain doesn't physically rotate a picture in your head, rather it just encodes the firing of neurons based on how you physically interact with the world. As long as the input is stable, your brain just maps it perfectly to reality. Section 5, how the brain sees. So how does this raw electrical data finally become this seamless reality we experience? We've officially left the eye, and we're now traveling deep into the nervous system. It all happens incredibly fast. In mere milliseconds, the electrical impulses race down the optic nerve. In split seconds, that data reaches a relay center called the lateral geniculate body. And then almost instantly, it's processed in the primary visual cortex at the very back of your brain. From here, the signals flow into even deeper regions to recognize contours, perceive motion, and identify colors. Honestly, your brain acts as the ultimate supercomputer here. It combines the two slightly different views from each eye into one single seamless three-dimensional picture. It merges those slightly different angles, fills in your blind spots, and adds complex shapes and motion. It even connects this visual data to your memory, which is how, instantly, you know you're looking at a dog and not a cat or a monkey. The right half of each eye sends data to the right hemisphere of the brain, and the left half to the left hemisphere, weaving it all together continuously. So I'll leave you with this quote. Your eyes are the lenses, but it's your brain that actually sees. It's a pretty provocative thought, right? If your eyes simply capture the light, but your brain is the thing painting the reality, what else is your mind quietly filtering out or altering entirely behind the scenes? Our vision is just this incredible partnership between the raw physics of light and the masterful interpretation of our minds. Thanks so much for joining me on this explainer, and remember, keep questioning what you see.